Hi everyone, I'm Benson Henry Jonathan. I'm here to speak on how to run agarose gel electrophoresis. Firstly, let me explain how agarose gel electrophoresis works. Agarose gel electrophoresis is a technique used to separate DNA fragments and other micromolecules such as RNA and proteins based on size and charge.
Here are the steps on how to run agarose gel electrophoresis.
The first step in DNA fragment separation is gel preparation, also known as gel casting. An appropriate amount of agarose powder is weighed out. For standard agarose gel electrophoresis, larger molecules are resolved better in low concentration gels, while smaller molecules separate better in high concentration gels. Once the agarose has been weighed, an appropriate buffer such as CAE or TBE is added. Agarose does not dissolve until it is heated. Therefore, the solution is heated for a few seconds in a microwave oven. Once the agarose is fully dissolved, the solution is cooled to about 55 degrees Celsius in a water bath. While the agarose solution is cooling, the electrophoresis tank containing a negative cathode and a positive anode electrode is prepared. For gel casting, a comb is placed in casting tray to create wells for sample loading. Casting dams are fixed to prevent the agarose solution from flowing out during settings. Once the agarose solution has cooled to about 55 degrees Celsius, ethidium bromide is added. Ethidium bromide is highly toxic and mutagenic, so it must be handled carefully in a fume hood. Ethidium bromide is an intercalating agent, meaning it itself is set between DNA base pairs. When exposed to UV light, it fluoresces, making DNA bands visible. The more DNA present, the brighter the band. After adding ethidium bromide, the agarose solution is poured into the casting tray and allowed to solidify at room temperature. Once the gel has solidified, the comb and casting dams are carefully removed. The gel is then submerged in CAE or TBE running buffer, which provides ion to carry current and maintain a stable pH. The next step is the preparation of DNA samples. A loading buffer is added to each sample. This buffer contains glycerol and dyes such as bromophenol blue. The glycerol increases the sample density so it sinks into the well, while the dye provides color to simplify loading and monitor the progress of electrophoresis. A DNA ladder containing DNA fragments of known length is loaded into the first well of the gel to serve as a size reference. After loading the marker, the DNA samples are placed into the remaining wells. The lid is then placed on the electrophoresis tank and an electric current is applied to pull the samples through the gel. Because of phosphate groups in their backbone, DNA molecules are negatively charged and therefore migrate towards the positive electrode. Since all DNA fragments have similar charge to mass ratio, smaller fragments move faster through the gel matrix, while larger fragments move more slowly. Due to the relative small molecule size of bromophenol blue, it migrates faster than DNA, and by optical control of the migrating colored band, the electrophoresis can be stopped before the dye. Once the DNA fragments have been separated, the gel is placed under UV light. Then the DNA fragments can be seen as bands. Each band contain a large number of DNA fragments of the same size that have all traveled as a group to the same position. By comparing the DNA bands of the samples to the DNA ladder, we can determine their appropriate sizes.
I'm Deborah David Ansah, a biochemistry student. I'm here to take you on the step-by-step procedures on how to run polyacrylamide gel electrophoresis. First, I'd like to explain what polyacrylamide gel electrophoresis is. It is a technique that is used to separate proteins and some nucleic acid based on their size and charges.
Hi, I'm Blessing. Here are the steps for running the gel.
The gel is made from polyacrylamide, which forms a porous matrix that acts like a molecular sieve. Before we begin, I'd like to take us on the gel preparation. A polyacrylamide gel is cast in a cassette with a comb that creates well for loading samples. Once the gel solidifies, the comb is removed. SDS page sample preparation. For SDS page, proteins are mixed with a denaturing agent like SDS to give them a uniform negative charge and denature them into linear chains. The sample are then often heated to ensure complete denaturation. Molecular weight marker. A marker with proteins of known size is prepared and loaded into one lane to help estimate the molecular weight of your sample.
Firstly, assemble the gel and chamber. Place the gel cassette into the electrophoresis chamber. Secure it in a place using the provided dips or a frame. Secondly, add buffer. Pour the running buffer into the inner and outer chambers of the tank, making sure the gel is fully submerged in the buffer. Thirdly, load the samples. Carefully load your samples and the molecular weight marker into the well using a macro pipette. Use a different tip for each sample. Fourthly, apply power. Connect the chamber to a power supply, matching the positive and the negative electrodes. The negative electrode is at the top and the positive is at the bottom of the SDS page. Run the gel. Turn on the power supply. The gel is run until a trackley dye such as bromophenol blue migrates to the bottom of the gel, and after the run, remove the gel. Disconnect the power supply, remove the gel cassette from the chamber, and carefully separate the gel from the glass plates. Stain the gel. To visualize the separated proteins, stain the gel with a dye like Kumasi Brilliant Blue or Silver Stain. Analyze your results. The gel can be imaged under UV light or with a scanner to visualize the protein band. The position of the bands can be compared to the molecular weight marker to estimate the size of the proteins in your samples.
In conclusion, gel electrophoresis separates molecules by size using electricity. Agarose gel have big pores that are used for separating RNA and DNA. pH have tiny acrylamides that are used for separating proteins.
Agarose contains horizontal DNA and UV light. Page contains vertical protein and Kumasi blue. We use this every day for checks, DNA fingerprints, and protein purification. Thank you for watching.